We talked about enzymes and how specific they are, and the idea that enzymes have activation sites. Those activation sites are pockets that actually grab a hold of the substrate and fit it like a lock and key, as it says here, but it also becomes a tight fit. This induced fit is the enzyme will change its shape slightly so that it really locks onto that substrate. But please understand that this locking on is reversible. Your book talks about a handshake, where you're going to reach and grab somebody's hand. Unbeknownst to you or beknownst to you, you actually change your shape of your hand slightly to fit into the template of theirs, lock onto it, shake hands, and then let go. You can think of that as that's how the enzyme works, how it shifts its shape so that that substrate will fit in, or substrates, if that's how that enzyme is working, and then reversibly let it go. And speaking of that, there are many ways that enzymes do work. They can work by making this microenvironment where substrates are more likely to bond, to form. And you have an image here of the catalytic cycle of sucrase, where you're taking the hydrolysis of sucrose into its components, the monosaccharides, glucose, and fructose. And what you're doing is taking a sucrose, which is a 2-unit sugar, holding it together in a enzyme and having that bond break. Or it can be backward. You can have the individual, or the other way around, not backward, glucose and fructose both being held together in that enzyme, and then allowing that bond to take place. So then you go from a monomer to at least a two-level sugar there, in terms of glucose. And so that's one way that they work. They also work because that active site of an amino acid, that R group, can temporarily bind to the substrate, and allow that then to allow the substrate to change or become what the enzyme is making that substrate become, or facilitating the substrate to become. Some of the things that affect enzymes: temperature, pH, concentration of the substrate, and so each enzyme is very specific. So it's not just enzymes work at a specific temperature. Each enzyme works at its own optimal temperature, and we consider this optimal conditions. And so you can see in this image, with regard to the graph on the upper left, at about 35, 37 degrees, this particular enzyme reaches its peak activity. Until that temperature, it's not at peak, and after that temperature, it's not at peak. And actually, the curve is quite steep. It reaches more and more peak level till it gets to the temperature, and then the temperature drops off pretty fast. The same is true with pH. Enzymes each work at their own more optimal pH. We have many that work at about pH of 7, because that's what water is, and you have many enzymes in water in your body, and inside the cell and outside the cell. But some enzymes in your body work at a pH of 1.5. Think of the stomach. So it depends on the specific enzyme. And please understand that if you take a specific enzyme and take it out of its optimal either temperature or pH, it can very possibly do what's known as denature it. An enzyme is a protein. Proteins are a particular shape. That denaturing can actually cause a change in the bonds and even break the bonds, and then the enzyme no longer has the original shape, and shape is very much a part of how an enzyme works, when referring to those substrates again. You also have this concept of a saturation point. So if we have 10 enzymes, each with an activation site, they're only going to be able to take care of 10 10 substrates at a time. Putting more substrates in, so putting 20 substrates in, we only have 10 enzymes, is not going to increase the rate that an enzyme is going to be able to be functioning. So there is a saturation point. A couple more concepts about enzymes to understand, and then we're finishing up this chapter. There are cofactors that assist enzymes, and cofactors are generally inorganic. We can think of vitamins, minerals, ions, minerals such as iron and zinc. They're necessary for the activity of the enzyme, and this can be for various groups. I mean, for various reasons. It can be because the active site needs, I guess it needs a cofactor to create that induced fit, or the cofactor actually helps create the active site. So these cofactors can be permanently attached to the enzyme. We call them prosthetics at that time, or they can be temporarily attached. So they can affect the active site, and that's what they're going to do, is affect the active site in some way, either with a permanent attachment or a reversible attachment. Enzymes can be inhibited. They can be inhibited in various different ways. One way is to be competitively inhibited. And so again, the activation site and that substrate coming in, well, if you actually have a chemical, which at this time, because it's acting as a competitive inhibitor, we're going to call it a competitive inhibitor, can actually bind to the activation site of the enzyme, and then the substrate can't bind. So this enzyme is not doing its job. That can be reversible, or that can be one that can be a covalent bond. If that covalent bond is the case, then this enzyme is not going to work any longer. It can also be non-competitive, and with non-competitive inhibitors, what happens is they bind to another site on the enzyme. And remember, it's all about shape with regard to that activation site, but binding to another site can actually change the shape of the activation site, because it's actually shifting the shape of the enzyme, and now that activation site no longer is recognized by the substrate, or the two are no longer able to do that induced fit. So inhibitors can work competitively or non-competitively. Examples are toxins and poisons, pesticides, and antibiotics. Then this idea of allosteric regulation brings up kind of another topic that we haven't talked about. Some enzymes are made up of complexes, not individual proteins, but complexes, and these complexes are oscillating. And this oscillation can be thought of as active form or inactive. So this movement or oscillation. And so you can have allosteric activators that stabilize the active form and make it more likely that substrates are going to bind. And that would basically speed up the, not the rate of the reaction, but how many of them take place. Or you can have this oscillation where you have an inactive form, and that then stabilizes the inactive form. So this oscillation back and forth and the enzyme activators and inhibitors can actually regulate how much of a substrate is made. We think of this with regard to ADP and ATP. ADP actually stabilizes an active form and creates more ATP to be formed, or inspires more ATP to be formed. A bulk of ATP, if you have a bulk of energy molecules, then you don't need more energy molecules made. Remember, all of this takes energy to make, so it needs to be regulated. And so you actually have, if you have a bulk of ATP, you have ADP actually slowing down that oscillation or the rate that, or the, you can think of it as the activation sites to allow more ATP to be made. And the next slide, with regard to, we do have a slide about the feedback inhibition. It might make it a little clearer for you. The allosteric regulations can also be cooperative, where you have the binding of one substrate molecule to an active site, and that then makes all of the active sites change their shape to allow more substrates to bond. And this one, the image is pretty clear. You have an inactive form. This oscillation is basically moving around. What the oscillation does is it makes the active site either available or not available. If we put a substrate in, it stabilizes that oscillation, so all of the bonding sites are out, and then more likely that the next substrate will be able to bond in there. That's actually the way that hemoglobin works. Your hemoglobin is not an enzyme. That's a molecule that carries oxygen in your blood. And once one oxygen binds in, it's more likely that the other, it actually carries 4 oxygen molecules. Once one binds in, it's more likely the other 3 sites will also get an oxygen binding in there. So the allosteric reactions can be activating or inactivating or cooperative. And then, as I said, the feedback inhibition here, because we can't just have these reactions going on willy-nilly. It is, remember, metabolism is the sum total of all your chemical reactions. All of those reactions are going on at different speeds at different times based on needs to remain in homeostasis. One of the biggest inhibition is the end product of the pathway. If you have plenty of the end product of the pathway, there is no reason for the pathway to continue acting. And so in this particular graphic, you have an activation site at the top, the purple enzyme. It is going to go through a series of steps to make isoleucine at the bottom. You have kind of the blue groups of balls, several levels of steps of enzyme activity. Then you get the kind of fuchsia or the salmon-colored groups of balls, the isoleucine. That then binds into an allosteric site. So binds into another site. It doesn't bind into the active site, but another site, and then changes the active site. So the end product actually regulates how much of the end product is made. Now, if the body uses the end product, uses up, then all of this is going to go forward again. The mitochondria, we're back to the mitochondria. It is where most of your metabolic pathways take place. And in terms of some enzymes act as structural components of membranes, some reside in specific organelles like the mitochondria, and the enzymes for cellular respiration are located in the mitochondria. In your body, it's not just the mitochondria that you have enzymes, but a good portion of them are in the mitochondria. You have many enzymes in the GI tract, that are released by the pancreas into the GI tract. You have some enzymes, I would say, mostly you have some enzymes in the liver, so you have areas where you have enzymes, but most of them are found in the mitochondria for this ATP production, but between there and the GI tract. And we are done with this chapter. We'll move on to chapter 9. Here's what you should be able to understand. At